Welcome, everyone, to our first webinar in a series exploring complex trait genetics. I am Alexander Arguello, Program Director at the National Human Genome Research Institute. This webinar is being recorded and will be posted on our series webpage and YouTube channel. The format of this series will be a presentation followed by a discussion with the moderator and then questions from the audience. Today, I am your moderator. You may submit your questions at any time in the Q&A, and we will get to them at the end. The goal of this series is to look at the cutting edge of the genetic architecture of complex traits. Instead of presenting standard talks on the latest research findings in a given topic, we really want to get to the edge of our understanding, to really explore that space where our current methods reach their limits and we don't know if our existing approaches are sufficient to give us the answers we seek. We hope we can challenge our assumptions and our preconceptions and think more deeply and broadly about really fundamental issues. Each webinar will focus on a different topic in complex trait genetics. And today we are starting by revisiting one of Richard Lewontin's more famous papers published 50 years ago. Our presenter will be Rasmus Winther, a philosopher of science, researcher, writer, educator, and explorer. He is professor of humanities at the University of California, Santa Cruz, and affiliate professor at the University of Copenhagen's Globe Institute. His latest book, Our Genes, is a philosophical perspective on human evolutionary genomics. He has called Richard Lewontin one of the most important evolutionary thinkers of the latter half of the 20th century. I thank you all for joining us. I look forward to the presentation and discussion, and I'll now turn it over to Rasmus.
Thank you, Xander, and thank you everyone for being here. And I am going to share this now. I want to thank you for being here. We all have many things to do, and I personally find Lewontin extremely interesting, and I hope I can convince you by the end of this talk that he still has many relevant things to tell us who care about statistics, genetics, philosophy. And I have this book out called Our Genes, where I discuss many of these matters, and Lewontin plays a centerpiece in it. Lewontin died a few years ago in 2021 on July 4th. And just to give, many of you may not have, I'm going to assume that some of you haven't heard of him, and I'm going to try to just say a few words about who he was and maybe what my relationship was with him. And then before I do, I'm also a diver. I love the oceans. And before I do a deep dive into one of his famous papers, 1974. I put up this slide because I wanted to just pull together some different obituaries to kind of show the impact. Now, yes, some of this is in very specialized places, like the Santa Fe Institute, obviously the Harvard Crimson, where he was a professor for much of his career, though not all of it. He was also at University of Chicago. And a third place, which I am blanking on, apologies right now. And I wrote the obituary in Science for the People, which was a journal that he was very closely involved with. Now, Lewontin is someone who interacted with a lot of people. And I'm not going to mention all the names in this diagram. I'm happy to answer for people who might be interested, but they include Stephen Jay Gould, hubby on the here, Marcus Feldman, his wife, Mary Jane, perhaps most famously, Richard Levins, who he co-wrote a book with. And then, of course, I also have the Engels and Marx. He was an avowed Marxist, although I will not have anything to say about that today. Hegel, Darwin, and so forth. That's Mark Feldman up at Stanford. And one of the quotes that I'm going to return to is he had this idea that context and interaction are of the essence. Now, again, for those of you who may not have heard of him, he did much of his theoretical work, which was very impressively influential in the 1960s and 1970s, and to a little lesser extent, 1980s. What happens in the 1980s is that he starts moving more, if one can say so, towards popularizing science, commenting on science, etc. But that's not to say that he didn't keep publishing in high-level places. He did. This is with Hubby and Lewontin, and I'll return to this paper down here. He wrote, I would say, one hardcore population genetics book, and that was A Genetic Basis of Evolutionary Change. And then he co-wrote with Richard Levins a book, and then I put in Biology's Ideology as one example of him popularizing science. Now, I have published quite a bit in different ways on Lewontin. Perhaps his slightly more famous paper is his paper on An Apportionment of Human Diversity, which is basically the paper that people who think about race and genetics like to cite. And it's a rather technical paper, but I really don't want to say very much about it. I did an exposition. I went through all the math. I recalculated everything. I explained everything in this paper of mine, Lewontin 72. And I have published on many of these topics. So what I'm here for today is to, besides tell you a bit about who Lewontin was, and you know, that I actually have something to say about Lewontin. You may not have heard of me before this announcement came out, is that he wrote what I take to be a conceptually absolutely thrilling, fascinating paper called The Analysis of Variance and the Analysis of Causes. Now, some of you, if you are in statistics, you have, of course, heard about analysis of variance and how that is used. Lewontin, while not a statistician, understood statistics rather, at least certainly the basic framework, but also a lot of the details of it at his time, shall we say, the 70s going into the 80s, he understood very well. And for example, if you ever want, and email me if you like, he has a beautiful article written in 1960 when he was a young man, where he goes through all the math of ANOVA and explains it, frankly, in a way that I think very few textbooks can live up to. So he understood how ANOVA works. Now, if you're a geneticist, the reason why you might care about this piece is that it really, to channel Xander, really tries to lay out some assumptions about how what are some of the methodological assumptions, some of the mathematical assumptions, some of the causal assumptions underlying analysis of variance. And I realized that I have a lot of text on some of these slides. I'm not going to be able to read all of it, but these are the 6 pieces, 6 sections of his article: introduction, discrimination of causes and analysis of causes, quantitative analysis of causes, norm of reaction, effect of additivity, purposes of analysis. Now, in the introduction, he says he's commenting on an article by Newton Morton and his colleagues that provide a detailed analytic critique of various estimates of heritability and components of variance for human phenotypes. You might wonder, by the way, why I'm reading and I'm quoting directly from the paper. I'm a philosopher of science as much as a geneticist, as much as an ecologist, et cetera. But sort of my core is philosophy and philosophy of science. And one thing we learn and one thing we love and one thing we think is really important is really try to give people the benefit of the doubt and work with the words and the concepts as they express them, but have that as your data. That's your data set, is the article. You got to read them carefully. And what he was forever worried about is a fallacy that a knowledge of the heritability of some trait in a population provides an index of the efficacy of environmental or clinical interventions in altering the trait, either in individuals or in the population as a whole. So he wants to analyze Newton Morton, but this paper is really an excuse to get at what ANOVA is trying to do and try to find some statistical and genetical pitfalls with it. So I would liken what follows to look rather closely at the problem of the analysis of causes in human genetics and to try to understand how the underlying model of this analysis molds our view of the real world. Like Levins, he was forever concerned with the relation between model and world and how we can often reify models into world. And that's something I spent a lot of time trying to make sense of in my book, When Maps Become the World. How does a theory or a map become the world? And when is that justified and when is it not? So in the second section, in a nutshell, he wants to distinguish two kinds of problems about causation. One is to discriminate when you have multiple alternatives and mutually exclusive causes which lie at the basis of some observed phenotype. For example, remember, he's writing in the 70s. This is the old problem of distinguishing major gene effects from polygenic effects. It's got to be one or the other, at least for one particular trait or one particular decomposition of that trait. So we can make a hypothesis that discriminates between two alternative causes of a human disorder. And he claims this is worth making if it can be done. Now, so he doesn't spend very much time on this problem in the rest of the paper when we can actually bring out alternative causes. Rather, he's much more concerned with the problem of analysis into separate elements of a number of causes that are interacting to produce a single result. What do you do when you have multiple causes of the same outcome? In particular, it is the problem of analyzing into separate components the interaction between environment and genotype in the determination of phenotype. So he gives this very nice metaphor. And I'm going to skip over some of it. If an event is the result of a joint operation of causative chains and the causes interact in any general accepted sense of the word, it becomes conceptually impossible to assign quantitative values to the causes. Only if the causes are utterly independent would we do so. Here's a beautiful metaphor. If two men lay bricks to build a wall, we may quite fairly measure their contributions by counting the number of bricks laid by each. But if one mixes the mortar and the other lays the bricks, it would be absurd to measure their relative quantitative contributions by measuring the volumes of bricks and of mortar. Likewise, if we cannot ask how much of an individual's height is a result of his genes and how much is a result of his environment, we can't do that. It's like the one person laying the bricks, the other one mixing the mortar. Well, what we can ask is what proportion of the deviation of his height from the population mean can be ascribed to deviation of environment from average environment and how much to the genetic value from mean genetic value. That is a reasonable question to ask. Now, I'm trying to go very quickly through the paper. He has one favorite thing. He thinks ANOVA isn't great, but he does think norms of reaction is great. And I'll get to that. I'll get to what they are visually in a second. Norms of reaction don't have the problem that the amount of environmental variance that appears depends upon the genotypic distribution, while the amount of genetic variance depends upon environmental distribution. Because that problem ANOVA has. That's why for ANOVA, the appearance of the separation of causes is a pure illusion. And ANOVA, which is based on the general linear model of statistics, it does not really affect the separation of causes of variation. And so it is a purely local description that doesn't have predictive reliability. I'll get back to this. I'm going to skip the point about sampling. Basically, he thinks what a norm of reaction tells the right causal and functional story that's summed up, whereas an ANOVA can't tell the causal and functional story. That's a picture, a visual of a real norm of reaction from an earlier paper by his PhD advisor, Dobzhansky. And viability up to 1 is on the vertical axis, the y-axis, and temperature and environmental variable is on the x-axis, the horizontal axis. And each of these lines represents some genotype. I'm not going to get into the details of what it does. It's actually an inversion. So that's just to give you a picture of a norm of reaction. That's an actual example. There's plenty of examples. There's a whole literature on this. Now, in the interest of time, I won't be able to go through every single one of these, but these are conceptually different norms of reaction that Lewontin draws. In this case, there is a net effect of environment, but there's no net effect of genotype. The average effect of genotype is the same across. In this case, there is no net effect of the environment. The average effect of the environment is the same across environments, but there is a net effect of genotype. I could go on. This is he meant to have as enzymatic. Enzymes work like this. This is, say, 2 genotypes for 2 enzymes. This is a so-called example of canalization. When over a broad range of environments, even for very different genotypes, you get approximately the same phenotype. And Conrad Waddington is famous in the 20th century for having really nailed this idea of canalization. There's work by, say, Melinda Fagan on this. And the bottom 2 are what's called additivity, and I'll get to that right now. There is one circumstance, he thinks, in which the analysis of variance can, in fact, estimate functional relationships. And rather than read the whole, this will be available online, and you can go look at the slides. I'm happy to share the slides with anyone who writes to me too. But additivity is when independently you get this, there is, when you add, as you add across environments or you add across genotypes, you add in a linear way. And this on the left is called near additivity. You're not quite adding in a linear way. So another way to put the point is independently of the environment, the difference between the 2 genotypes is the same. Or independently of genotype, the effect of the environment is the same on phenotype. So that's the case of additivity, and that's a really important point that we will get to in a minute. Now, he does end the paper by saying the purposes of these analyses are different. The analysis of causes in human genetics is to provide us with the basic knowledge we require for correct schemes of environmental modification and intervention. Look, he's not saying that you can't find causation in human genetics, but he thinks that's a much more mechanistic and a much more experimental way of doing it. It's intervention. Like you actually have to do something. He thinks statistical analyses aren't a great way to do this and don't really give you causes. So he also thinks that knowledge of norms of reaction can predict the demographic and public health consequences of certain massive environmental changes. And ANOVA can do neither of these because its results are a unique function of the present distribution of environment and genotypes. I am going to sum all of this up in my next slide, 2 slides, okay? I'm going to skip this middle one. He ends the piece by saying, this is the last 2 sentences. He always has very provocative, controversial last couple sentences. His race paper, the last 2 sentences, the last paragraph is brilliant, if nothing else for its provocative nature. He ends this paper, however, in view of the terrible mischief that has been done by confusing the spatiotemporal local analysis of variance with the global analysis of causes, I suggest that we stop the endless search for better methods of estimating useless quantities. There are plenty of real problems. He thinks ANOVA is really only good for estimating the rate of selection in humans and to reconstruct, in some cases, the past selective history of the species. Okay, so let's sum up what Lewontin is trying to say. First, he's saying, these are the global theses I take. 3 global theses in the paper. ANOVA is local and contextual. It doesn't support inferences to new context or new data. You have to redo everything. You have to redo the data because there may be all these hidden variables you hadn't taken account of. There may be interacting epistatic effects you haven't taken account of, but I'm getting ahead of myself. ANOVA is local and contextual. There is no prediction possible from ANOVA. ANOVA explains At best, and only via variance components, differences of variation rather than actual traits. There is no explanation of particular cases. And ANOVA fails a priori, that is beforehand, to identify variance components or causes. Why? Partitioning of the world is not pre-given. We have to make assumptions about what the hell are we measuring? Oh, genes and environment. But that's an assumption that was made in the 19th century, starting with, not even with Darwin, arguably, but this is a long story, perhaps Galton with nature nurture, but I don't think that's the right way to frame this. But no matter what, buying into genes versus environment is what I call a partitioning frame with a lot of assumptions behind it. And ANOVA and regression and GWAS, which I'll also get to in a minute, cannot, except under highly idealized assumptions, give us what I'm calling a causal picture of the world. ANOVA cannot, says Lewontin, give us a causal picture of the world. Look, I can sit here and do a song and dance about how great he is, and in 10 minutes try to sum this up. Nothing will beat it, will beat you sitting down and spending an hour of your life with this paper, if nothing else, skimming it. It's beautifully written. No one can accuse Lewontin of being boring or a poor writer, okay? So I'm trying my darn best to summarize it, but it's not easy. Okay. Some local theses is that the main effect variance component, like the gene main effects or the environmental main effects, they influence and condition each other. This is a very strange thought, but it's actually true. And it leads to consequences built in the assumptions and speaks to the limits of why you can't keep using ANOVA. And the reason it's getting dark here, I'm sitting in Denmark. That's why it's getting dark. ANOVA can only provide a proper causal story or functional relationship when additivity holds. Those are local theses. Now, this concludes me having summed up the paper. I hope Xander, who kindly invited me, is satisfied enough with an almost impossible task to sum up a paper, and I hope some of you have at least learned something about it. But I'm going to give you now we're going to deep dive into the consequences. And why is Lewontin even writing this? I'm going to give you 3 context slides of the additive paradigm. And I can't spend too much time on any one slide, but you can ask me all you want. Lewontin quotes this piece by Arthur Jensen, who's a psychologist, who writes a very controversial paper, and he's one of the first so-called hereditarians. And his paper is called "How Much Can We Boost IQ and Scholastic Achievement?" One thing that's pretty evident from having read that paper is he's pretty close to the additive paradigm. This is a figure literally from the paper. Oh, I am so sorry. Oh, you know what? It should have. Oh, God. And it didn't come out here. I cut it wrong. The y-axis literally says intelligence or IQ or something. It definitely says that. And I somehow, in cutting it, putting it in, that got left out. But for the record, the y-axis says IQ or intelligence. It's figure 11 of this paper. And then he has favorableness of the environment. And look at his norms of reaction. They're near additive. They're all kind of go up. So independently of the environment, genotype D is always better than genotype C, is always better than genotype B, is always better than genotype A. However, they each get a little bit better over time. And that's what he thought individuals were. Some individuals are just smarter than others, independently of the environment, but give them a better teaching environment and they will get better. And he made similar arguments about races, and other people have made the argument, but Lewontin is writing this article to beat that point up. Why? Because he's saying you don't know what the normal reaction is. You don't know whether they're going to cross or how they cross. And frankly, genotype isn't going to tell you very much here either because of all the statistical quandaries that I've just told you about. Here's another background that Lewontin really liked and was influenced by Sewell Wright. And everyone at the time knew who R.A. Fisher was. Again, there's a lot of political matters currently about R.A. Fisher one could talk about, but I think regardless, we can bracket, I want to just bracket that, because regardless of that, there's zero doubt that in the 20th century, Fisher was extremely influential in the rise of statistics and in formulating many statistics. He invented ANOVA. Let me say that again. R.A. Fisher invented analysis of variance in papers in the teens and 20s. Okay? And he assumed roughly the relation between dependent and independent explanatory variables is linear, normal distribution of errors with constant variance, cross-variable explanatory impotence. So if you had different explanatory variables, they didn't interact with one another. He ignored outliers, and he didn't call it this, that came later. But he built the basis for the general linear models of statistics, which assume additivity and abstracting away from error and interaction. There's a kind of ceteris paribus, all other things being equal, and there's a lot of ceteris absentibus, all other things being absent, going on in a randomization, the randomized additive worldview, evolutionarily too, of someone like Fisher. Which leads me to go from race and IQ to statistics to genetics. I'm singling out Richard Dawkins because in his Selfish Gene, he calls Sir Ronald Fisher the greatest biologist of the 20th century. That's a quote in the Selfish Gene. And behind the Selfish Gene, and I've written extensively on this, I think the best place, if you want 3 pages on this, I wrote a review of a book on the gene's eye view by O'Kane that appeared in Evolution that compares and contrasts Lewontin with Dawkins. It's Hardy-Weinberg assumptions gone wild. Infinitely large populations, panmixia, no fitness interaction or epistasis among genes, no too little. Dawkins accepts phenotypic plasticity, but he doesn't make a big deal out of it. No little phenotypic plasticity and the fullness of time. And then the assumption in Hardy-Weinberg you do include under this picture is the potency of artificial and natural selection. It's the same kind of additive worldview. And I understand now that I am running somewhat out of time. One can make all the beautiful PowerPoints you want, and you run out of time sometimes. Lewontin always insisted on epistasis and gene interaction, that there's always a limit, that genes are always interacting. And you can't tell a full causal story about about evolution and changes in gene frequencies unless you include as many, basically you have to include all genes. And the last chapter of this book is called The Genome as the Unit of Selection. He thinks of the entire genome as the unit of selection because every gene is like interacting with every other gene for fitness in all kinds of weird and unpredictable ways. So the selection of the chromosome as a whole is the overriding determinant of allelic frequencies. I'm going to read this one quote. The fitness at a single locus ripped from its interactive context is about as relevant to real problems of evolutionary genetics as a study of the psychology of individuals isolated from their social context is to an understanding of humans' political evolution. In both cases, context and interaction are not simply second-order effects like interactive effects in ANOVA to be superimposed on a primary monadic analysis. Context and interaction are of the essence. Another way he strikes back, although he let go of this later in his life, but he did think that evolution happened at many levels. So there's an agency of natural selection of molecules, cells, populations, species, and communities. And he's also kind of well known for really having tried to nail the 3 conditions of evolution, that there's variation, differential fitness, and heritability. So the questions related to this audience, this public, are questions about complexity, interaction, and emergence. How do we best handle complexity in our statistical and genetic models? We have many limited models. There's all kinds of ceteris paribus and ceteris subsentibus assumptions. But what about data-rich and computation-heavy analysis? Do they solve Lewontin's questions? I argue no. Can we restart our theory? Then there's also questions about what is interaction? What interacts with what? Is interaction fundamental? Friend and colleague Helen Longino, she has a beautiful paper, Interaction, a Case for Ontological Pluralism, you can check out. And then, is nature basically hierarchical, or does it single out and or bottom out? A beautiful book on this topic is William Wimsatt and his work over the many decades, Re-engineering Philosophy for Limited Beings. These are two philosophers of science I have just mentioned. This is a thought experiment from my book, where I contrast three kinds of universes. And I think okay, I'm just going to read just a second quote of each of these. In its essentials, a clockwork universe is taken to be deterministic, orderly and regular across space and time, potentially long-lasting, simple, and potentially totally explainable in that all causes can be postulated and identified, and their effects fully mapped out. In contrast, a soup universe is stochastic and chancy. Do read the last chapter of my book. I promise you, it ain't so bad. A soup universe is stochastic and chancy, disorderly, likely short-lived and unstable, so utterly complex that any instruments and measurement protocols attempting to measure it are useless, and unexplainable because regular and robust causes do not seem to exist. What I try to then do is to say that, in a way, the general linear model in statistics is trying to combine the determinism of a clockwork universe and kind of making assumptions about the universe and all the interacting phenomena in the world and mechanisms and processes in the world acting in a very clockwork fashion, but then trying to use that to domesticate the soupiness, which the world also has. So in a conscious universe, however, both clockwork and soup elements operate. The GLM imposes elements of clockwork linear law onto soup-like chance to yield insights and far-reaching meanings. And I spent a lot of time in that chapter, that last chapter, trying to make sense of how statistics would work in our conscious universe. That's literally the title. Statistics in a Conscious Universe is the title of the last chapter. No, sorry. The Conscious Universe, Genes and Complex Systems. Oh yeah, apropos. The section is called, yes, here. Genes in the Conscious Universe, Genes and Complex Systems. Okay. So very apropos this webinar. I'm going to switch gears in the last couple minutes I have to causal analysis. XKCD is always a good place to go look. I used to think correlation implied causation, then I took a statistics class. Now I don't know. Well, says the friend, sounds like the class helped. Well, maybe. I trust I don't have to explain that. Okay. All right, Lewontin, you're making all these claims about the weaknesses of ANOVA and this and that. Yes, you are right that if we don't know very much about the range of environments, and if we don't have much data and this and that, it's very local and it's probably very contextual, and we need to keep doing the experiments and doing the data analysis. But what do we do today when we have hundreds of thousands of individuals, many of which are sampled, not perhaps for the whole, not often for the whole genome, but for a lot of the genome, and we have highly effective computational power? So, for example, GWAS, and also we can use fine mapping. We can use network analysis. We can randomize Mendelianly. We can use machine learning to get polygenic scores and to improve statistical power, etc. And we can detect interactions. The more cases you have, the more you can detect interactions. And all the footnotes, all the articles are there. And can we use GWAS to develop drugs? Well, there are examples where we kind of already have, or we've developed sorry, I'm just getting slightly tired and slightly a little concerned about the time, but I will finish the whole thing within my 40 minutes allotted. And can we use GWAS to prove that certain drugs or to predict drugs? And yes, you can. So does that belie Lewontin's analysis? I'm going to say no. And here I'm going to bring in the interest of time, bring in for 4 minutes some philosophy. It is still the case that even if all the evidence we have, there's still a lot of underdetermination of theory by evidence. There are still many alternative models and many alternative hypotheses that can fit or somewhat fit with some massaging the different evidence. I mean, the picture might be closer to this one on the bottom right, that the evidence, look, the evidence itself, no one agrees 100% on all the evidence. No one agrees on all the data analysis. I spend a good chunk of chapters 4 and 5 in this book showing how different modeling methodologies for human genetic diversity actually don't give very different results depending on what assumptions. Think of structure, the structure program, and then variance decomposition methods. So people don't even quite agree on the evidence, and the models can still are underdetermined. And we might know a lot. So yes, it's true. If Lewontin is here with this dot, we're way beyond Lewontin in the present state of inquiry. But we certainly haven't looked at all practically possible circumstances with experiments and surveys, say. We haven't looked at all naturally possible circumstances. What, we're going to look at ancient environmental DNA? We could. Maybe we're going to look at ancient environmental DNA and look at human genetic diversity across time. But what about all logically possible circumstances? Could we look at different Earths and different, I know that's a bit out there, but the point is Lewontin still holds some water if certainly the statistical assumptions, the critique of the statistical assumptions he makes are well known to statisticians. So, but they'll say, we just have to be careful with the modeling. Yes, we do have to be careful with the modeling and the analysis, but even so, there could be multiple models. Like, what if you're outside of the environment? What if you haven't looked at a certain gene locus or a certain target gene that actually has a lot of epistatic interaction with other genes? We would need the full picture. What about total evidence, as Carnap said? Would Lewontin then be wrong if we had all the total evidence? I'm going to punt on that question. You can ask me again. I think, yes, there's a part of me that wants to say then he would be wrong, but there's a part of me that would say, but then you still need to know the mechanistic information. But I'm punting on that. I'm going to skip induction. I'm going to try to motivate this rather than explain it. Explain what GRU is. When you look at a norm of reaction, it is just, what about all the points that we still haven't measured? And what about all the points that are not on that line, but that are the line that we can't measure all possible environments and all possible genes? So what about lines that we haven't measured? And what about extrapolation and interpolation, to also use cartographic language? We don't know. Yes, we can make assumptions, linear assumptions, but then we're making linear assumptions again. We can't know whether the line is going to be the same. And I invite you to look at, and this goes back to an idea, Nelson Goodman, another Harvard professor, literally colleagues with Lewontin for many decades, about GRU. Green before a certain time, blue after that time. And everything we've examined today that is green, we don't know whether it's green or GRU because we haven't reached that point in the future yet. That's to motivate. There's a lot of skepticism still here. And in fact, I would say in causal analysis, there is. In philosophy, there's so many views about cause. There are too many views. I can't read. But I do think even if you care only or primarily about genetics, causation, and so forth, it might behoove you to at least think about why is so many different views about causation over the last since the scientific revolution, or let alone since Plato and Aristotle? And I would kind of group them into three. Look, there are more than three for sure, and this is not the best grouping, but either cause is in the mind, like Kant thought, that cause was kind of a primordial category that was built in. It's an a priori synthetic category, he called it, that was built into the mind, without which we couldn't even make sense of the disparate phenomena. Phenomena wouldn't make sense unless we already had the category of causation in our mind. Go read some Kant, or at least go read some commentators on Kant. Kant is notoriously hard to read. Then you have Australian realists like David Armstrong, who thought that causation is something in the world. Causal relations are in the world, and my guess is that most scientists think something like this. But then we still have the Kantian questions, and we still have the questions of how to recognize what the world even is, and isn't there some framing happening by humans and by inquirers? You can take Bertrand Russell, who in 1912 wrote a famous paper where he basically took a very skeptical attitude. He starts the paper by saying, this is not his words, but paraphrasing him, he just says, look, the notion of cause has so many meanings, it's basically hopeless, and it doesn't really do very much in physics anyway. And this is a very common view, actually, that cause doesn't do very much in physics. Go read some Ernst Mach, or if you're interested, Imitation of Rigor, who's a philosopher, Mark Wilson at Pittsburgh. Really interesting book about the notion of cause and how people who think about philosophy of physics have got it all wrong. And he is a philosopher of physics. Well, he's a philosopher in general. And there are also many views in statistics. There are many forms of causal. There are many studies of the mathematics of causal inference. Yes, many of these totally go beyond anything Lewontin is writing about in the paper. But I think a lot of the assumptions of linearity, it's not something I think, it's something they admit. Like Imbens and Rubin start the whole book by talking about these various assumptions they have to make. There's a lot of work that goes into statistics where you're making all kinds of assumptions. And Lewontin was just kind of, shall we say, saddened or kind of frustrated with all the assumptions that were made in statistics, but then we reify models that are based on those assumptions. That was in a way, his primary concern. Thank you, Xander, for inviting me. Thank you, audience, for being here. And of course, also thank you to Lewontin, who wrote this very lovely, to Rasmus, who has helped me think about important issues in his copy of Genetic Basis of Evolutionary Change. Thank you. I'm done.
Thank you, Rasmus. That was fascinating, interesting. I learned a lot. Now we'll sort of get into a little discussion between yourself and me, and then we'll also look at some of the questions that ended up in the Q&A. So I want to start something light. So you've clearly written on about Lewontin over the years. What about him specifically drew you to him versus someone like his colleague Stephen Jay Gould or Dawkins that you mentioned, or even E.O. Wilson, which were some of his contemporaries? What was it about Lewontin that sort of got you really excited?
That's a lovely question. I don't want to go on and on about it. So many of my teachers at Stanford themselves had been influenced or worked with Lewontin, had either been his postdoc, or Mark Feldman had collaborated with him. Peter Godfrey Smith had been a postdoc. There were other people who were grad students who had worked with him. And the more I've read of I just really love his combination of technical acuity, philosophical or conceptual rigor, empirical knowledge, and, if I may say so, political savviness, which, politics is very messy, I know, but at least I appreciated his consistency, and I mean, I think his big heart. Yeah. So it's not the best answer, but Stephen Jay Gould, I've read most of his essays up until the 90s. I loved him too, but well, I'll be honest with you. Well, I've met both of them very relatively early on in my career, and Gould wasn't very nice. Dick was very nice. I don't know how to say, I shouldn't say these things, but there, I've said it, it's on the record, but yeah.
It goes to show that collegiality goes a long way in
Yes,
forming
collegiality goes,
relationships. Yeah.
yes. And
So I
that's
want to
also
talk.
part of the reason he had so many students. He had like, I mean, it was a hive. Like his lab at Harvard was the hive. It was a hive. There were always people coming in and out. I mean, literally, they were making honey. I mean, they were like, it's really interesting stuff. I mean, it's nourishing. And he accepted so many different kinds of people. I mean, also across the political spectrum, believe it or not. And also just people who were more philosophical, people who cared more about politics and race, and people who were just like literally just whatever sometimes, what is it in English, the nose to the grindstone or whatever you say, like the tech people who just doing the technical stuff. Yeah, which is great.
I want to get into a little bit of the relationship between science and philosophy. A lot of our audience are scientists, students, postdocs, early career researchers, and I'm sure they're thinking right now, and there's a question in the Q&A reflecting this, what is the practical implications about thinking philosophically about these types of questions? It's not like a hiring committee or a tenure committee is going to be asking people, are you a realist or instrumentalist, right? Why should scientists think more philosophically in general?
Yeah. So obviously, yeah, a fascinating question that I've thought for decades about. Can you do so? I fundamentally think so. Oh God, how do I do this? I'm not sure you can do science. You can do technical science without philosophy, maybe. Yeah, and you can discover new things maybe without philosophy. But I think behind every big theoretical conceptual revolution, I do think there's some philosophy in science. But maybe more interestingly, if you care about understanding what statistics is really trying to say or do, let me be a little more specific because science is a big thing. And, I love physics and, I mean, have like very impressive physics friends who, professors in fancy places. And, I don't even understand a millionth of what they do. And they claim to me that philosophy is important, but why should, if you're a geneticist or a statistician or an ecologist, care about philosophy? Because for one, you're always going to care about concepts. So concepts like population, like interaction, like frequency. And yes, they can be operationalized as definitions, but in a way, they are philosophical concepts. And there's going to be as many different meanings of population, frequency, and interaction as there are smart scientists and philosophers thinking about this. And how will it help you in your everyday work? In this book, I spend a good amount of time writing about what I call assumption archaeology. Every theory, every model, every statistical methodology, every network analysis model has all kinds of assumptions. Undigging those assumptions or diving deep into those assumptions is going to require, ultimately, it's a conceptual exercise. Do you have to be a trained philosopher? No. But I would argue that you're being a philosopher. You're acting like a philosopher. You're as if you were a philosopher when you're thinking about concepts or thinking about assumptions or thinking about what is it that I'm really asking here?
Yeah, I often hear it said that every scientist is an implicit philosopher, but it's only the really rigorous scientists that explicitly acknowledge their sort of philosophical stances. Um, so I wanted to get to something you mentioned in your talk about partition framing, right? And so again, so this idea about genes and environment has been around for a long time. And usually the critique has been that we just focus too much on the genetics, not enough on the environment. You know, the genes are discrete and easy to measure. The environment's amorphous and messy. But now with the collection of more sort of phenotypic data, there's more interest in collecting information about the environment. But again, that's still presupposes this partitioning, right, of genes or environment. Do you think there is a way to move beyond that, even now, as we still sort of take that framing in terms of the approaches and the data that we collect?
Xander, I have to apologize to you because I didn't catch the last two sentences because I'm noticing there's a Q&A. And I know I was also told by Jake not to look at the Q&A, but tell a kid not to look and they will look. Sorry, I feel, yeah, I'm a little bit of a kid's always. I just, the world is endlessly fascinating. So can you repeat the last two
It was
sentences?
just, oh, can we move beyond the partition framing of like genes versus environment, or genes
Yeah,
and environment, or sometimes you talk they both matter, but that still seems like not satisfying as a response, right?
I agree. So I think we already always have, because you just have to look at the different disciplines of biology. I mean, it's relatively few disciplines. I'm going to make a very provocative claim. I'm not even sure it's true, but it's the very few disciplines of biology that actually only look, I mean, or fundamentally accept gene versus environment. Look, cell biology. Yeah, yeah. So I'm thinking this out loud. You're asking me a hard question. What about ecology? I mean, we have to be specific, right? I mean, if you care about development and individuals, and you care about outcome of a particular kind of process, like developmental processes, or if you care about evolution, then yes, then many biologists will have a gene environment partitioning frame. But I think there are like areas like biophysics or membrane biophysics, or ecology, fisheries biology, et cetera, where gene environment is just not that important, a partitioning frame, but then it's falling outside of the scope. I do think we can, even for those fields that care about genes and environments, then or marine biology doesn't really use it, then we can think a pet peeve of mine is that the environment as a environment as a category covers too much. What are you talking about when you talk about environment? And what Lewontin always insisted on, where's the organism? Where is the individual in all of this? And genes and environment are always interacting at many levels too. So you have to look at the cell level, you have to look at the tissue level. So that is left out of the story too, when you're doing a completely abstractivist, what William James called a vicious abstractionism of genes and environment. It becomes nothing but genes and environment, but there's all these, even in development, there are all these levels. So we definitely need to specify environment much more, and we need to specify genes, and we need to specify in which context genes act. And I know we don't have that much time, so sorry, I'll stop that answer now.
Okay, so I just want to get to a couple questions in the Q&A. The first one is sort of a practical question. Someone asked, what are sort of the pitfalls of sort of assuming this linear and additive framework in studying genetic effects? And they're looking for a specific example, something that, if taking that framing is going to lead you astray.
Well, I mean, so if you don't take into account epistasis, you're leaving out a lot of, I wish I had this. I've learned from my teaching, never try to conjure up a slide that you have in a PowerPoint hidden somewhere. So I'm not going to do it. But if you have multiple loci interacting, but let's say you have 3 loci interacting, and you've only really caught 2, and you're only doing the dynamics of 2 of the loci, you don't know what activity the third loci, whatever the shapes say to development, may happen if you care about it, whatever the effects on phenotype of those two loci. You can try to make predictions based on, oh, now I have these two settings for these two loci, and you can try to make predictions. But if you didn't know that the third loci had always been on while you were making the experiment, then you just don't know what happens when it's off. Then the prediction isn't going to hold for those two loci. I mean, I'm really just trying to make a very simple case. And this happens a lot in genetics with epistasis. So as long as if you accept gene interaction happens, and whether it's mechanistic epistasis or probabilistic epistasis, this does happen. Sorry, I'll stop there.
We'll get sort of one last question from the Q&A. And so Lewontin famously, wore his political ideology on his sleeve. To what extent do you think science can be separated from one sort of ideologies and other domains of social cultural life? Do you think the approach Lewontin took is one that can be scaled up, or should people be more careful in sort of mixing their science and their other beliefs?
Okay, so this is, sorry, I'm going to be even more provocative. Climate change, sorry. We live at a time of where my politics on my sleeve, we live in a breakdown times. And even when I talk to my ecologist colleagues, et cetera, marine biologists, et cetera, whether it's at Copenhagen, whether it's at Santa Cruz, whether it's at Stanford, et cetera, everyone is most people are somewhat worried to very worried, personally. But publicly, no, et cetera. So I think that's one case. And what about studies of human genetic diversity? This is too big a question. I'd like to say, look, the problem is I want to say that people should be allowed to wear their politics on their sleeve and then not be persecuted by their universities. Sorry for saying, but it does happen to some extent. Happens especially with climate action. And I just, I think we're seeing a shift in the relation between politics and science. And I think we're also seeing a bit of a, God, I have to be careful. But like I'm with Lewontin on a lot of his politics, but I do believe in freedom of speech. I do think if there's, sorry for saying, if there is someone who wants to interpret genetics in a different way, and as long as they can make good arguments, the data, et cetera, et cetera, et cetera, they have a right to be protected to make those claims. But I think the political landscape is different now than it was for sure when Lewontin was publishing. Maybe it shifted more to the left in universities. It's hard to say, but it's a big question.
All right. I want to thank our presenter, Rasmus Winther, again for that illuminating presentation. This is being recorded and will be available on our website and YouTube channel.
I want to answer one QA. Can you disagree with someone politically and still trust their science? Yes.
Thank you. Yes. So go to our series webpage for the next upcoming seminar. They'll happen every couple months or so. So look there for updates. Thank you again, and everyone enjoy the rest of your day.
Thank you.